And I think it's really important because when you think about feeding everybody, and yes, we do use poultry and beef, you have to look at it in a sustainability aspect too, of just how much land and how much greenhouse gases that they're using. And then really think about, okay, how do we minimize that? How do we still feed everybody? And then that's when these alternative proteins come into play.
Well, welcome to the Pet Food Science Podcast. I am here today and we're going to talk about sustainability. Meredith, you're our expert guest of the day, and I'm very pleased to have you. I'd like you to just introduce yourself and tell us a little bit about your background and how you got to be interested in pet food science.
All right. Well, thank you so much for having me on today. My background, where to begin? It's kind of a little extensive. So I got my bachelor's at the University of Illinois around 2021. I completed it. And during that time, I was actually interested in equine reproduction. So I worked on the University of Illinois horse farm, worked with their standardbreds, helped with stallion collections, looked at the semen longevity. I also helped with the reproduction aspects of just artificial insemination, things like that. And then I also was an undergraduate research assistant for Dr. Kevin Klein, and we looked at semen motility in stallions. And then about my junior year, I realized that I wanted to look further into nutrition, realized U of I didn't have an equine nutrition program, fell in love with Dr. Kelly Swanson's nutrition, animal nutrition courses, reached out to him, wanted to be in his lab, and he generously took me in as a non-thesis student for graduate student. And so there I had this mealworm project that I was working on, and I fell in love with alternative proteins and what that meant for the industry, what that meant for me as a researcher. So I really wanted to stay on and get my thesis, and then eventually now become a PhD student and look farther into alternative proteins.
84 million times a day, pets eat meals with ingredients from Trow Nutrition. We bring together the science of ingredients, nutrition, and blending to unleash possibilities for pet food brands. Premixes are just the start. Turn to Trow for higher inclusion ingredients too, like proteins and carbohydrates, and highly sensitive ingredients like probiotics. With our palatants and base blends, you can feel confident about what comes in our bags and goes in yours. Learn more at TrowNutritionPets.com.
No, that's good. Thank you. Starting with this whole idea, I think, for the person who might be listening to this, Meredith, might be going, well, wait a minute. I thought, pets, they pretty much always eat chicken or beef or pork, and you're talking about mealworms. I mean, this is a long stretch. Can you tell us why that might be important, for a pet, someone feeding a pet?
So, for my understanding is the world is going to be growing in population. We're going to keep getting animals, and we're going to need to have a bunch of food to feed all these people and these animals. And right now, our biggest issue is, are we going to run out? And what does that look like? And are there other alternatives that we can use to help add to the nutrition or the diets for our pet foods? And that's when we started looking at insects, or I guess you can also say that there's fermentable plant proteins that people are looking at right now. And then what that means in terms of pet food. And I think it's really important because when you think about feeding everybody, and yes, we do use poultry and beef, you have to look at it in a sustainability aspect too, of just how much land and how much greenhouse gases that they're using. And then really think about, okay, how do we minimize that? How do we still feed everybody? And then that's when these alternative proteins come into play.
Well, that makes sense. On the one hand, the pet food industry has been really significant in using the alternative products that people don't want to eat. So they are producing, beef and cattle, chicken, many different muscle protein sources or animal protein sources. And then a part of that protein is not used by people, but it's available for pet food because people don't want it. But this then is a supplement that's really probably uniquely going to be used in pet food. Is that right? So this would be designed and the other thing that's so clear, and I know you're going to talk about it some, but I'd like to kind of introduce it, is that from a nutrition perspective, the nutrients in the food are not based on the ingredients. So if we have lysine, if we have a digestible protein source, we as humans or the pet, it doesn't care if it's a chicken lysine or a corn lysine or a meal lysine. It's the amino acid that matters. So Meredith, I'm going to ask you, you've been involved with this sustainable protein source for pets. And how does that get measured? What do you do to say, it's acceptable, it's even good? What are the processes you follow?
So basically, to look at the protein quality, we first assess the indispensable amino acid digestibilities. And so basically from there, we want obviously greater than 90, but greater than 80% is pretty good. So then we'll consider that a good protein quality ingredient. And then from there, just because we know that obviously amino acid digestibility is super important so that the host can utilize those amino acids once the protein is consumed. We want to look at the digestible, indispensable amino acid scores of that specific protein ingredient. So what that means for me is I'm looking at if all of the indispensable amino acids exceed 100% for their scores, meaning that, let's say, lysine, for example, is under 100%. That's going to be the limiting.
Well, what we need to know, can I interrupt you? We need to know a little bit for the audience about the denominator. So 100% lysine is what, maybe 2%, 5% of the total mealworm. What is that 100%?
So basically, that 100% is from the 1 gram of the protein that we're looking at. So if you look at the amino acid in grams divided by the amino acid in that specific ingredient, and you times it by 100, you're going to get the score.
Meredith, you're using a standardized profile to establish that percentage, right? So you've got amino acid in grams is what you said, but it's an amino acid. Say, for example, you desired a pet food protein that was 5% lysine. If your mealworm was then over that 5%, then that would be over 100%. Is that correct?
Yeah. Mm-hmm. Yes.
So my question then is, okay, so you've got, that's an analytical analysis, right?
Yes.
So that's done with an amino acid analyzer, pretty complicated machine. You're doing that at University of Illinois. Can you tell me about how that analysis works?
So we actually send off our analysis to the University of Missouri. And then we get that data back. And then from there, I look at the dispensable, indispensable amino acid scores from that amino acid digestibility data.
So do you start out with the sort of chemical score of the ingredient then? So you say, independent of digestibility, this looks like a pretty good ingredient. It's got maybe all over 100%. I want 2% lysine, I've got 2.5%. I want 0.3% tryptophan, I've got 0.35. It's all looking marvelous. But then your next question, as I understand it, is you're going to find out what does the animal get out of that? Because it could be there but not used, right? It could be not digested. So tell me how you do that digestibility.
So basically, when we look at the digestibility, we're going to take the amino acid in the test protein, and we're going to look at the percent, and then we're going to reference the same amino acid in that reference protein, if I'm understanding your question correctly.
Well, I'm trying to get us to talk together about establishing the value to the pet. Now, digestibility of amino acids is kind of a little
Mhm.
bit tricky, right? Because the microbiota can chew those proteins up and make the amino acids of their choice. And so, if you use a sort of normal whole tract
Oh,
digestibility,
okay.
which is great for nitrogen, because nitrogen is neither created or destroyed in this whole event, but it's really kind of rotten for a
Oh, okay.
indispensable amino acid, tryptophan, lysine, or whatever. What are you doing that says, okay, I know that this is really being used by the pet, not kind of being recreated by the microbe here?
I understand your question now. Okay, so we use cystectomized roosters.
That's what I wanted to talk about.
Yes.
So this is
Okay.
a model, right? So we don't do that surgery for pets, and
No.
we're just going to step aside. We're not doing any surgical procedure for pets, but we have these roosters. They live a very happy life, right? They're roosters. They're doing
Yes.
very well, but they have this function in life, which is they really model this. Why does that model work? Why do you use that model?
So we remove the twin cecum in the rooster. So that mimics the fermentation of those microbes that are eating the protein. So we remove that. So essentially, we're looking at the ileal digestibility, let's say, of a dog or a cat. And then by removing those twin ceca, we can mimic that track so that when from the moment that the rooster consumes that ingredient to the moment that it's excreted, we don't have any microbiota interference.
Yes, that's really critical, isn't it? Because otherwise we could say, well, look, it looks like lysine, for example, is not being totally being like, it's not that 80-85% we
Mhm.
want. Maybe it's down 50-60%, but we wouldn't know that in a dog because maybe those microbes are saying, look, I got all the alanine, this dispensable amino acid I want. I think I should make some lysine. And then the lysine is we wouldn't know. We wouldn't know if the pet was using it or not. So this is a method to say, okay, we've got not only the nitrogen being absorbed, but we've got the specific amino acids of concern that we're really interested in absorbing. That's
Yes,
making
correct.
sense. So,
Yes, correct.
mealworms. What is it about mealworms that brought that into the space versus maybe cricket meal or whatever other alternative protein source might be interesting? Was there a company that came to you, or was that your source? Tell me about that.
Usually, a company will sponsor one of our studies in the lab. And for this sake, the company from Europe came to my lab um, and asked us to look at these mealworms specifically. We've looked at black soldier fly in the past based on another sponsor's request. But yeah, so it pretty much just depends on the sponsor's needs and what they want us to look at.
So this is a good industry and academic partnership, right? So we see a need. It's a valuable need for pets, valuable for the sustainability of our environment. These are good things. And so connecting together, they came to the University of Illinois into your space and said, we'd like to do this analysis. Now you completed the analysis. Sounds like you did this assay with the roosters to say that it was digestible. Did you do anything with dogs and cats?
I didn't personally know. I know that the recent article that just came out, they did a 6 month long study with dogs to test for the total tract macronutrient digestibility and also just to look at safety measures while using this ingredient in a diet. But no, unfortunately, I did not get to work with dogs with
No,
this.
I understand. The thing that interests me is that there's so many steps in bringing this idea into fruition. So the first idea is, okay, there's an ingredient supplier, they've got an ingredient of interest, they come to the university, you do some testing, but you still can't actually put that in pet food, right? So tell
Mhm.
me about this whole AFCO umbrella that recently was maybe hurdle is not really a hurdle because they're protecting pets too, but it's something that you have to get over. Can you tell me about how that worked? There's a recent meeting, I think.
So in regards to the use of mealworms in the dog food, you mean? Okay.
Yes, yes.
So yeah, so they have to undergo the 6 month long study that I just had mentioned to make sure that all of The dogs remained healthy in the study. I don't know specifically if they did look at digestibility, but that would have been a great measure to see how much of that ingredient is being utilized for the amino acids. But yes, it's a giant hurdle to jump over. There is a lot of compliance that you have to work with, specifically with the FDA, making sure that it gets generally recognized as safe or GRAS approved first. And then the AFCO can come in and then look at the regulatory statements and then make their definition and approve it for use in pet food. So there is a lot of moving components surrounding whether or not the ingredient gets approved.
Well, and the good thing is that protects the pet, right? So that this is done so that if someone uses an ingredient, there's a well-standard scientific background to say this is safe, efficacious for a particular purpose. In your case, meeting the protein requirement of the pet. Seems like, you sort of declare success and go home. Is there anything left with the mealworm thing that you'd be interested in, or is it complete?
I believe that I might be getting some, I guess, different formulation of the ingredient, like whether or not it's in a slurry, whether or not it's processing differences, and looking at if that has an impact on the nutrient digestibility and overall protein quality. So that might be in the near future, but for me, I'm moving towards looking at fermented plant products. And so unfortunately, we'll see. It's up in the air.
Yeah, no, I understand. And I guess what I'm thinking is I would like the audience to hear, and it's just that secondary thing is that now it is an acceptable, approved, and safe ingredient to use. However, there's really a lot to know about ingredients. And I love your statement of processing because, my experience has been it's not always just what's in the food, but how it's made that matters. So that's really a good next step. A lot of the world lives on soybeans, and pets can do very well in soybeans. Some of the things, of course, we know about fermentation is some of the anti-digestive components of soybeans can be removed by fermentation, or as you know, by processing in other ways. But you're interested in looking at a fermented soybean product, and tell us again just what you're going to do with that to evaluate value for pets.
So I'm just going to look at the protein quality and the amino acid digestibility of that ingredient.
So when you do that, you sort of don't get the opportunity to the broader spectrum. Nutrition, you're going to get a PhD in nutrition, not in digestibility.
Yes.
And so what would you recommend? Maybe it's not you who are doing it, but what would you recommend maybe if you beat the hurdles, right? You said we've got these standards, we want to be above 80%. My guess is this product is going to be above 90%. It's going to be very digestible. But you beat those hurdles, say, okay, here's the next step. What would you do? What would you recommend to evaluate this ingredient?
I would recommend putting it into a diet, whether that probably an extruded kibble diet. And then from there, look at the apparent total tract digestibility of that diet with that ingredient included in it. And then compare that specific diet to, let's say, commercial chicken and rice diet to see how that compares.
So you'd be interested in the protein digestibility of those two things. You'd probably be taking the chicken out and putting the fermented soybean ingredient in. What else would you look at, though? Again, I'm interested in thinking aloud with you about this, this whole body effect. You talked about maybe a probiotic in there. Dr. Swanson's lab is very involved with many things that would touch this. What would you you're in the lab, you've got the free scope. What would you do?
Look in terms of probiotic.
I mean, you said that this ingredient was going to have a probiotic in it, if I understood. There was a fermented soybean, and then they were adding a probiotic, and you'd like to know the value of this. Probably not going to see it in your customized rooster. Did I say that correctly?
Yes.
The probiotic doesn't have a chance, right? It's a non-microbial assay. So you want to test that now. What would you do?
So I would look at, again, I would put it into a diet. I would feed it to dogs for an extensive period amount of time, probably like 4 months. And then I would probably have those dogs go under some sort of stress to then see if that probiotic has an effect on the immune response of those animals, and then look at the microbiota from there, and then see if there is a shift depending on animals that are on a placebo effect versus the probiotic, and how well that immune system played an effect on those animals. And then I would also, again, look at the cortisol levels just to see like how much stress they then endured from the transport, and then probably make a conclusion as to whether or not that probiotic was helpful in those hosts or not.
So it's fascinating. I think one thing that we've seen in pet nutrition, and really this is a biology, but this microbe immune response interaction has been fascinating. What do you think some measures, what would you do to measure immune response, or maybe even to try and look at that crosstalk? You talked about cortisol. This is a hormone that sort of reflects stress in the pet, but what other things might you look for the crosstalk for how those microbes and how that pet and the immune response, the host, if you will, is responding to those changes in the gut?
Yeah, so I would also look at cytokines and chemokines in the host. We're doing ELISA. We draw blood. We then get the serum from that blood, and we run ELISA analysis to look at all of those cells and how they're interacting, or like their levels within the blood. And that would give us a good indication of whether or not their immune system is functioning like properly, or if anything at an optimal rate, depending on if they had undergone stress or not.
So one of the things that interests me, and you talked about this ELISA technology, you want to just tell us a little bit what that stands for and how you might use it in this. There's a lot of things influencing the immune system. So you're touching on this spectrum of things. How would you take this to that step of saying, wow, I'm seeing this whole body effect for the pets?
So right now, because my research is solely focused on protein and I haven't really jumped into using or working with ELISAs yet, I can't really speak to exactly what it stands for and like all of the breakdown components of it, because I haven't run the process myself yet.
Sure, that's okay. Well, I guess I was thinking aloud, and it's a great thing about this podcast. We can just think aloud together. The thing that fascinates me is this. So ELISA stands for enzyme linked immune sorbent assay, right? So it's a way to. These things are very low concentration in the blood, and it's a way of amplifying that, and you get a much better analysis than you could do if you're trying to run it on a size exclusion chromatography or something is going to fail. The thing that I'm fascinated with, and I just hope you can tell us a little bit more as you think about your career in pet food, is that digestibility is a standard and functionally wildly important. It will get your ingredient of interest a long way down that you want to use it. But in the end, if I'm a nutritionist putting it in a pet food, I've got this spectrum of protein sources. I'm really pleased with chicken. I really like soybean in the context of a whole nutrition. I wouldn't want a canine or a feline food that was only protein source was soy. But you have to work it together so that we have what we call complementation. And I'm just trying to get you to think with me about how that complementation could be measured to the benefit of the pet. So we talked about immune system. You've talked about inflammation with the cytokines, because you have these pro-inflammatory cytokines. We do a lot with ingredient evaluation. Any other thoughts on that? What would you as you see your career going forward, what would you be interested in? Do you want to focus really on digestibility and those foundational measurements, or would you like to see a breadth further than that?
So I do look at the microbiota on looking at obviously digestibility, but then looking at the microbiota and like looking at DNA when we take samples from our pets, and then seeing exactly which type of diets or diet formats, like different processing methods, have an influence on our gut microbiota for dogs. And depending on what they're fed, or like, let's say, yeah, like the different formats, if you're looking at a fresh pet food, frozen raw diet versus an extruded kibble, and how does that play an effect to the bacteria that's in our gut, and then what bacteria is more pronounced. And for example, I've done a study in the past where I looked at raw-based meat diets as compared to a raw, like a red meat diet, extruded kibble, and saw results saying that there's more proteolytic bacteria that's infesting in the gut when you're eating a raw-based diet. And what exactly does that mean for the pet? Is it sustainable for the pet if it's being fed for a long period of time? I know that proteolytic bacteria cause a lot of fermentation in the gut, which can also cause bloat, which is not super exciting or comfortable for the host. Things like that. I really want to dive into understanding a little bit more about what specific bacteria does, how we can influence that. Does it make a difference when we change diets frequently with, like, for example, all these new pet food companies are coming out with feed your pet chicken and rice one day, feed your pet beef and rice, feed your pet fresh diet, and then seeing how that influences. I think that's where the industry is going, and I think it would be really interesting to see how that impacts the gut.
Yeah, thank you. I think that will be interesting too. A good space for probably a lot of nutritional expertise and evaluation to come.
Mm-hmm.
What do you think? I always like to ask people, Meredith, and I hope you would sort of think about together as you look at Dr. Swanson has done so well with creating an environment for successful teams. And,
yes.
You can observe that it's just true. What do you see in that from your perspective that maybe has made those teams successful? What's the ingredients that drew you in and that made it seem valuable?
I would say the passion from my teammates in the lab, and just like the communication. Communication is so important when you're on a team. You have to be able to obviously communicate what's going on. If you have any questions, people are always there to help. That's super important, especially when we're doing research, we're trying to figure it out, we're trying to hopefully get good results. So it's good to like collaborate and bounce off your team, see what they have any input. I would say, again, passion, being driven, being organized. Organization is huge in terms of, obviously, because we're graduate students, we have to take classes and stuff like that. But being organized, having a plan, and just overall, being a good team member, I would say.
Okay. Well, that's pretty exciting. Sounds like an exciting place to be.
Yeah, it is.
What do you see as you see sort of kind of maybe your thoughts in just because I'm interested, maybe someone watching will be interested too. Is there a good chance that you can be in your area when you graduate? What's your career, sort of goals and aspirations?
I would say, so I'm going to be prelimming, hopefully. So I'm still in the thick of all of my PhD work, but I'm hopefully going to be prelimming and finishing my PhD in 2025. And based off in Champaign right now, I'm from the northwest suburbs of Chicago, grew up there, but I have a broadened horizon, I guess. I don't want to limit myself. I really do want to be involved with alternative protein still. When I go to conferences and I meet other companies, it's super interesting to see what they're working on. They're innovators. That's really exciting for me. And just, I guess, being open-minded, I also do really like diet formulation. So we'll see where that takes me.
Okay. Well, thank you. I've enjoyed having this chat. So why don't you summarize for the audience, if you will, your experience to date with this idea that maybe there are ingredients out there that we're not using fully and how they might benefit pet nutrition?
to basically summarize everything, there are a lot of new and up and coming alternative protein ingredients. It's really important to focus on the protein quality, looking at the digestibility, and then putting them into a diet. And then to be open-minded also as a consumer or person buying this pet food, to maybe be open-minded to having insects included in your pet's food, or soybean, or just have it be plant-based in general. But I would stress that it's like super important to just overall make sure that your pet is getting its optimal nutrition that it needs.
Okay. Well, thank you. I've enjoyed chatting. I wish you good success as you go through your program. Well, I think you've seen just a great success in your first AFCO-approved ingredient that now is really widely available for pet nutritionists to enhance their foods, and use compatibility to use that ingredient with other ingredients to reach that optimum nutrition you've spoken of. So pretty exciting. Thanks for being with the Pet Food Science podcast today. And I've sure enjoyed getting to know you a little bit and hearing about your exciting research.
Thank you so much for talking with me today.